Okay, it's Patty Pesavento's turn now at UC Davis. Patty is the new chairman of pathology at UC Davis Veterinary College. Congratulations.
Or kuda, or so sorry.
Yeah, more work, more meetings, and more, you know, all that.
No, it's all good. It's all good. Hey,
sure
I want to introduce Dr. Willard, Kevin Willard. You guys know him, especially you NIH people, because this is really his story. Although Jerry knows the work that the story that started this model, but the model is all Kevin. So I don't think we're going to be nearly as generous as Elijah was in let me share my screen, but in describing what we're seeing or telling the story. We'll tell the story afterwards if is that kosher for this session?
Whatever you like.
Ah, right. All right. So again, I defer to Kevin for all things mice. And I stick to my furry little natural disease where I can't statistically prove anything. So this is the case. Kevin, you want to say anything about the overview?
Yeah, just, you know, you can see here, there's this, you know, irregularly circumscribed, pretty well demarcated mass, you know, densely populated cells, central area of necrosis. You can see in the lower left-hand side that it extends, you know, essentially from the area of the lateral ventricles, kind of obliterates the lateral ventricles, and then extends rostrally into the olfactory bulb. But stays within one hemisphere, you know, doesn't cross over to the other side.
All key information, you guys, for the story. I'll come a little closer to the pattern. And anybody comment on what they're seeing while we go?
The brain pathology expert besides Kevin. Anyone on here? Do we have any? We should call on people, but anyway.
It's a neoplasm.
Full credit. So, you know, there's not a lot of distinctive features for the cells. You know, they have a pretty high nuclear cytoplasm ratio. There's not a lot of patterns that you see, but robust mitotic activity that's present. You know, it does have necrosis, but lacks this sort of pseudopalisading around it that you see with higher grade glial tumors. And then, you know, pretty decent anesokaryosis, at least, you know, ranging from the round, you know, more vesiculate, round nuclei with vesiculate chromatin to, you know, more ovoid. Some even have a little tapered end.
I'm going to go over to the edge so that you can see its relationship with the surrounding brain, and then also what the reaction or lack of reaction thereof.
So it contains these plump cells with eosinophilic cytoplasm on the long sections, right?
Correct.
And so that pink stuff must be. Oh, you can talk about what's in those cells.
You want the answer? Is that what you're saying?
Oh, the answer that comes later. Yeah, okay. Well, anyway, so also I can lead the discussion here. So you have the whole sarcomatous differential schwannoma, sarcoma, meningioma. You know, a whole bunch of things that could be possible. Anyone strong feelings about anything?
To be fair, and I'm going to put this out there. To be fair, this tumor was one that defied all odds of classification, and a whole lot of neuropathologists weighed in, I'm going to say very passionately, to be professional, about what this was.
Okay. Well, it's very easy to know the answer and then expect that from other people, and you don't get it. So,
well,
seldom wrong, never in doubt.
Well,
actually, Sandy's the tumor pathologist #1, and Bob Cardiff. Hey, Bob, what do you think? He's not a brain guy, though.
No.
I vote that it is a neural-based lesion, not a generic sarcoma. That's kind of as far as I go.
You know, I love that you have to make that distinction. Since I only do natural disease, if I see a tumor in the brain, it's a brain-based lesion. But you guys have to consider all possibilities.
Right.
So are there any other lesions that we should know about?
No, this is it in this, and I'll go to the story because for time. And what we're gonna do is I'm gonna show you one other slide while I'm here, and then I'll go to the PowerPoint. And this slide is in situ hybridization for raccoon polyomavirus.
The subpolyoma.
So yeah, so it's a polyoma, but and so you can see the okay, I'm going to say addiction or the association of the virus with the tumor is very, very high in these mice. And then I'll tell the story, and we can get back to these slides if we want to analyze them. So, so for those of you who don't know the story, it was a kind of an amazing story that started in 2013 about where wildlife that comes in naturally to our diagnostic system. Among the raccoons that were screened for typical things like rabies and distemper, we had a really remarkable percentage. At one point, 20% of those that were neurologic from these centers coming in with a brain tumor. And that brain tumor was canonically in the same place. So picture this, you guys, from basically one county, because we had a rehab center. All the raccoons that were being submitted and screened for, 20% of them that were neurologic had a tumor that was located sort of centrally around the olfactory tract of the brain, sometimes extended into the nasal cavity, often extended into the nasal cavity, and often pushed back the brainstem like you see here, causing the neurologic signs that were picked up by the rehab center, which were circling and lack of aggression. I mean, if you can pick up and cuddle a raccoon, that's abnormal. And this was happening at a remarkable rate, in their opinion.
Can have rabies?
Rabies in raccoons is very, we haven't had a rabies case in raccoons in California for a good 30 years, Jerry. We don't really recognize it at all because its indigenous cycle is skunks and bats out here, and not the raccoons. That being said, we assume it's possible, and one of the reasons they're screening the neurologic is to make sure that that's not happening. But typically, a neurologic raccoon would be bromethylin toxicity or distemper. And so having such a high percentage having a tumor canonically in the same place, and having a tumor at all before you're hit by a car and you're a raccoon, is very unusual. So that made us stand up and consider, and me as a virologist stand up and consider what could be driving this. And we considered toxins, since it's in the olfactory tract, and whether or not there was some kind of problem that way. We considered, and I of course considered viruses. And so with a degenerate primer strategy, we picked up a novel polyoma virus that was present in 100% of the tumors. Now again, because we have this problem with a dividing cell population, we were kind of like, well, is it just a piggybacking on a dividing cell population? We don't know. But we did a lot of other work before Kevin nailed this one. First of all, remember, you guys, I just want you to recognize that a pretty high burden of human cancer is associated with an infectious pathogen. And I use that term associated very, very carefully, because proving causality is a high bar, a very high bar, and takes, for in the case of papillomaviruses, was a 50+ year journey, scientific journey. So starting with roadkill wasn't the easiest way to go about that, but that's what we had. And so we took it on as best we could. I'm going to give you guys some background on this, which is that the viral load in the tumors was significantly higher than the viral load in all of the other tissues. And Elijah, you see this urine thing right here? We looked with Ish at all of these organs that were positive by quantitative PCR. And the kidneys of those raccoons look exactly with just a tubule here and there, and nephritis, the chronic nephritis that's no longer associated with the virus, exactly like your case, like no difference at all. Inclusions in some of them, not in other ones, but it was present in a lot of tissues at really very low persistent levels, if you will, in these raccoons, but it was very, very high in the tumors. And when we had cases that had metastases, and in this case, you're looking at naturally occurring raccoon, metastasis to the liver, and then here is the RNA scope-ish of raccoon polyoma associated with that metastasis. So by how we define it, that viral genome and the large T antigen, which I'll get into, or the oncogenic portion of that virus, is faithfully segregated with the metastasis. And so that's some persuasive argument that this is a viral caused tumor rather than a tumor with virus.
Is there infection of normal tissue in those animals?
Yeah, the kidney in these animals, like I said, but only just spotty, like once in a while a hepatocyte, once in a while a renal tubule, epithelial, proximal tubular cell. I think we saw it in salivary gland in one case, even though several were positive. But what you'd expect from a very clever virus that wants to live out your lifetime and not be immune recognized, it has strategies to not cause inflammation, in most animals. And so we don't see them. We miss them.
There's no mutation in the natural virus or something?
Is this a mutation?
No, no, yeah.
Not as far as we know, Jerry. The sequence from the tissues and the sequence from the tumors was the same.
No, but for example, it exists in these raccoons for how long in nature, and then suddenly they develop.
Yeah, don't know. We don't have a raccoon colony, so can't tell you that. But our assumption is that it's a persistent infection. We know that if we look across the country and Canada, that the seroprevalence of exposure is the same as it is for Merkel cell polyoma virus or JT virus or the other polyomas that in humans we know are persistent and sporadically can cause disease. So the story is similar.
Oh, I see. Okay. Hey, Jim, look, in Massachusetts, you got some of those raccoons. You better go get them.
Well, we'll go trapping.
Yeah, or just pick them up off the road. Why not? It's cheap.
Right.
So remember that polyoma has an oncogenic portion, which is a single, it really is just two genes, that are differentially spliced. So you have the oncogenic large T antigen or tumor antigen, which is, as we know, goes far towards sufficiency for transformation because it's the reason through the poliovirus contamination with SV40. It is the very reason why we discovered so many tumor suppressors, P53, retinoblastoma, all of those were discovered via polyoma. So it's got a rich and very important history in virology. And then our structural genes over here. And I just want to point out before Kevin takes over with the xenograft model, which is what you guys were seeing, that when we look at the transcriptome of these tumors, this is the oncogenic transcript over here, very highly transcribed. In fact, top 5% of the transcripts, including all the host transcripts, top 5% in those tumors. Whereas these are the structural genes, and somehow, despite the fact that the virus is episomal, and we showed that it's not integrated, somehow LT is highly transcribed over VP. So another nail in the coffin of oncogenesis. And then we were left with these questions, which is, what the heck is the target cell for transformation? How is LT expression higher than VP? And is RAC-PYV really oncogenic? And this is where Dr. Willard came in spades. He did a lot of the work above too, but this is where this xenotransplant model and his cell culture system came into play. So I'll let you take over, Kevin.
Yeah, so after hearing Patty talk at ACVP about this right before I joined as faculty here at Davis, the anatomic location of these tumors struck me in that they're always in the olfactory bulb, and given I like some of the chat, especially from Enrico, whose differential was PNET, for primitive neuroectodermal tumor, because it's that's what I was thinking too in some of this, based off the morphology. And the location, as you can see in the lower left-hand side, using rat as a model worm. But you can see that that this is the location of the rostral migratory stream, which is proliferating neural precursors that arise from the subventricular zone and then migrate and divide as they go into and are sort of matriculate into the olfactory bulb. And so the location of the tumor and the primitive sort of nature of the cell morphology made me wonder whether or not it could be arising from these long-lived neural precursor cells in the subventricular zone, and then as they proliferate in the rostral migratory stream, then you get expression of LT from the RAC polyomavirus, and you get tumor formation. So we can go to the next one.
Yeah.
And we can just click all the thanks. So this rostral migratory stream, again, it's this really defined pathway where these neural precursors first proliferate and then ultimately differentiate. And so we isolated some cells from these tumors and grew them similar to how we would grow mammalian neural stem cells. Here, these were taken from a dog, but these neural progenitor cells have bipotential differentiation, where they can differentiate along either neuronal or glial lineages. You can go to the next one. And so we were able to isolate cells from these primary tumors and grow them in serum-free conditions, and we had cells grow. And most of this work is off of a couple of cases here, but we were able to grow these alongside neural stem cells from raccoons and these renal tubular epithelial cells as well. But we're going to mostly focus on the tumor-derived cells. And so we grew the raccoon tumor cells in the serum-derived or serum-free culture medium, and you can see that up top that they express markers of neural stem cell biology, the intermediate filament protein nestin, and then the transcription factor SOX2, and you can see that their expression pattern is very similar to olfactory bulb neural progenitor cells from a normal raccoon, and then you can see that they express markers of both neuronal 2J1 or beta-3 tubulin and astrocytic GFAP proteins in the below image. And so they have all the features of a neural precursor cell. Go to the next one.
Yeah.
And so we tried to look at xenograft models for a couple of reasons, not the least of which was to prove that they were tumorigenic, and then to see whether or not the expression of RAC-PYV-LT antigen was maintained in these tumors. And so we injected 20,000 cells just anterior to the lateral ventricle in mice and allowed the tumors to develop naturally. And we got tumors in 6 out of 6 mice, and the tumor location and the histology was maintained in all of the mice. You can see the median survival time is about 2 months or so. And then, ultimately looking at, again, ISH for the T antigen, we can see that the viral protein expression is maintained in the xenograft tumors in a very similar manner to the primary tumor.
And I'll chime in here that I was really fascinated with, because these are heavily immunosuppressed mice, I was really fascinated with whether or not what Kevin was transplanting was the tumor in raccoon cells or the virus that caused a tumor in the mouse. And so he did do host analysis of the genome. And the cells that you're seeing in the non-skid gamma xenotransplant are raccoon cells, host cells in that mouse. I think that's it.
That's it.
Did you try and bias the experiment to do the opposite, to introduce virus? And you mentioned, Patty, there's a very high bar to prove causality, but one of those proofs would be isolation of virus, readministration, even to a raccoon, much less a mouse, to see if you recapitulate disease from the virus.
Yeah, so the problem with that, Sandy, I mean, aside from trying to keep
aside from that's really hard.
Aside from the fact that keeping a colony of bored raccoons busy, is that the truth is that we don't know why these raccoons were suddenly, and really we've been scanning raccoons for a long time, and the East Coast and Fox can attest, they look at rabies raccoons all the time. This was a cluster of tumors, and we don't know what predisposed these animals. It could have been a transient immunosuppression, a heavy distemper season, something like that. And so in order to recapitulate what happened naturally, I don't even know what I'd do. I'd need immunosuppressed raccoons or some kind of corticosteroid treatment, or if I did just wild type.
Well, I mean, I agree that's a good hypothesis, but I think you could also just super challenge, right? So super challenge may be sufficient. But
a raccoon.
It's hard to know without doing, and you could do it obviously to mice, but it may not be cross-infective. I don't know. There's a lot to know. It would be informative for human disease too, as you say, with Merkel cell polyoma.
Yeah.
Which is also kind of a mystery. Like, how did you get that? Is that just passed around and most people have no problem with it? And why do some people have a problem with it? It's still a mystery.
Yep. Yeah.
What is the mechanism for neoplasia in large T antigen? So we can't really confuse this with the usual polyoma, which is polyoma middle T, that's all over the place in genetically engineered mice.
So, Bob, it's a good point what you make. And when I say large T, I'm probably being careless about using that term because there's so much alternate transcription, and we didn't distinguish among, we saw that there was a whole lot of differential transcription and that a sort of middle T existed, but we didn't actually distinguish those because the cases that we had, again, most of them weren't fresh. So doing that in a raccoon was impossible, because remember, we're hearing about these literally as roadkill. So the opportunity we had was in Kevin's model. And in that case, Kevin, you looked at the transcriptome in those. And I think we did some look to show that there was a full length LT transcript, but there was also many other variations of that T, splice variations.
So I'll just help fill in the molecular biology gaps for the audience. The large T antigen we know will bind to and inactivate P53 and RB, both of which are tumor suppressors. The polyoma middle T antigen functions as a molecular mimic of ERB2. It activates PI3K, SHIC, and SARC, and the PI3K pathway, and for example, P10 loss in brain tumors is common in humans, actually. So this is a common molecular pathway that I think is also directly pertinent to more widespread disease than just this raccoon outbreak.
Okay, I'm sorry to interrupt, but it's getting late, even though we don't have to stop exactly on time. So any last comments, Kevin or Patty? No, you're okay? Great, well, thanks for that.